this and I'll try to post these. My goal is I've recorded the last couple lectures. I will try, or at least portions of them, I'll try to get those up on YouTube today and send you guys links. No guarantee there because sometimes these take a while to generate, but I'll try to get on that this afternoon for you guys, right? So when we talk about generation time and population doubling, right? You know, from a medical standpoint, there's a lot of reasons that we care because contamination, right, specifically public health issues, right, exist because of how quickly these can grow. And remember, this is probably not shocking to you, when we talk about growth of bacteria, right, we're not talking about individual cell size, we're talking about population size, all right? So, well, and this doesn't apply to you because you guys aren't in lab, so you can't remember your broth inoculations here. But exponential growth, right, is going to start with, or in this case, this is just an example. We're starting with 10 cells. And I'm saying the population doubles every 20 minutes, right? So every 20 minutes, right, a bacteria can divide. So we have this handy dandy little formula here, all right? And so this is the total number of bacteria. This is the number of bacteria that you start with, all right? And this is the number of generations that occur, right, within an hour, right? So if I told you that a bacteria doubles in size or population, there's a generation, right, every 20 minutes, or doubling in population every 20 minutes, how many generations are there in 1 hour? 3, yes, perfect. All right, there are 3. So 3 generations every hour, right? So in this case, you would say 10 times 2 to the third. So if you started with 10 bacteria in 1 hour, how many bacteria would you have, Trent?
Math is not my strong suit, and I'm still trying to write all this down, so give me a second.
Okay. Someone help Trent out. Laura?
Sorry. Would it be 80?
It would be 80, right? 2 to the third. 2 times 2 times 2 is 8, right? Times 10, so that gives you your 80. And so you can see here, this is just showing you kind of what those generation times look like, right? So this is 3 generations that we did. You know, here would be 4 generations, 5, 6, 7, right? And you can just see that number kind of go up, right? And again, the generation time for different bacteria differs. Some bacteria maybe could have a generation in 20 minutes. Some of them might be a generation every 2 hours, right? So you just have to kind of be aware, pay attention, right? You know, to that time. And I would always say, you know, the generation time is 20 minutes. How many generations or how many bacteria are present in an hour, right? I'm not going to have you guess generation times. I would kind of give you a clue or hint as to figure that out. But you can see how population could grow really, really quickly, right? And typically, if we're talking about like an inoculation in a lab setting, you guys are not starting with 10 bacteria. You could be starting with millions of bacteria. So if you had millions of bacteria and you let them go for an hour, right, you can see how the population can just kind of take off, all right? If I were to ask a question like this on the exam, I, of course, will give you the formula. You will be, right, responsible for knowing what these different components of the formula stand for, though, okay? So here, this is really important, right? I love this chart, all right? You see me? Olivia, Amanda, I love this chart, all right? And this chart shows, right, kind of the life of a bacteria, right? What's going on during its lifetime. And so in this case, one of the things that I want you to assume here is that this is kind of maybe a perfect situation. This is a laboratory setting where it's growing on a plate or growing in a tube of broth, right? Where it has kind of an abundant source of nutrients. So the first thing that happens for a lot of bacteria is this thing called the lag phase, all right? And during the lag phase, you can see here, this line is flat. That means there's no growth, right? And the reason there's no growth is because you have enzyme synthesis, right? And these enzymes that you're synthesizing are going to help you utilize the nutrient source that's present in your environment, all right? So basically, all right, the bacteria is preparing itself to acquire as much energy as it can, right? It wants to be prepared, right? So there's no growth here. It is gearing up, right? They're gearing up to get ready to have a big feast, right? But they're not there yet. You then have log phase or exponential phase, all right? So you can see here, this is a sharp peak up, okay? And so these guys grow really rapidly. There's a lot of rapid growth. And again, rapid growth for bacteria means rapid division, right? They're just growing, growing, growing. The number of cells just keeps on going, 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 right? You then have a point where you use all of the nutrients, right, that are available. But, all right, the bacteria aren't dead yet. So think about it, right? If when you have breakfast and then let's say you wait till lunch to eat again, you don't automatically die, right? You've eaten all the food that's in front of you and you're fine for a while. But in this case, right, there's no more food. So if I gave you breakfast, that's the last thing you ate, you'd be fine for a while, but eventually you would die, right? You'd need more food. And so bacteria go through a death phase. This death phase is actually relatively short and brief. All right. It is also exponential, right? So it's a pretty quick decline. But you see here that the decline, right, kind of levels off a little bit. So this phase of prolonged decline, all right, we have adaptation to adversity. So bacteria in here start to adapt, right? Start to go through microevolution. They gain the ability to use other nutrients that are in the broth. They even, in some cases, are scavenging off of dead bacteria. All right. And this can go on for days, months, years. All right. Depending on the size of the culture. If, let's see, Amanda, if you were trying to treat a patient with antibiotics, what phase do you think you'd want to try to catch the bacteria in?
In the log phase,
And why?
because that's when they're undergoing their cell division. So I would think that's when they would be most vulnerable.
Right. And so I totally like that, right? If you have an antibiotic that's targeting cell wall synthesis, well, they're building a lot of their cell wall during this phase, right? Because they're dividing. So that's a good time to target them. Even if you're not targeting the cell wall, you're targeting other metabolic activities of these bacteria. All right. And when you target other metabolic activities of this bacteria, right, they are highest during this phase. Okay. Another thing to consider, and we don't really get to go over this, is when you're doing research or when you're testing bacteria, what phase do you not want them in? And I guess you could tell me 2 phases you don't want them in. What 2 phases do you not want bacteria in if you're doing research on them, or if you're trying to test them or figure things out about them?
The death phase or death of prolonged decline.
Perfect, right? So the death phase, Rachel, you got me there, right? If they're dead, you can't test them. It just doesn't work. What would be the concern in the phase of prolonged decline from a research standpoint, or if you're trying to test them on a plate that is selective? What do you think? Why would that be an issue there?
they could adapt to like live off of other things on the plate.
Okay, that could be interesting, right? So I'll kind of run with that. That helps me out, right? So Rachel's saying that maybe, right, in this phase of prolonged decline, some type of selective media would not have allowed the growth of a certain bacteria before that phase of prolonged decline. But the bacteria changes, right? Its metabolism is now different. And so the tests that you would have used before to identify the bacteria may no longer work because they're different, all right? And so if we were in lab, right, one of the things that I would tell you is, hey, when you're testing bacteria, the best place to have them is right here. This gives you the most accurate information. If you're testing over here in the phase of prolonged decline, oh, they've changed, right? They may not respond to the test the same way that they did before, and that would change your results. And so one of the things in a lab setting, and I actually do a relatively poor job at this, I'll admit it, is you have to keep your bacteria fed. They burn through nutrients relatively quickly, and if you let them get over here, it's going to be a lot harder to characterize and classify them. The other seed that I want to plant in your head, right, and get it in there now, is I want you to start to think, right, and this table, the table that was the end of our last PowerPoint that looked over prokaryotic cells and eukaryotic cells, was talking about some of the differences between DNA and RNA relatively briefly. And so what I want you to start to be thinking about is what allows bacteria to undergo this exponential growth? What is different about their ability to replicate, different about their ability to make RNA, different about their ability to make proteins, right? That allows them to grow so much more rapidly than our cells can grow, right? This growth chart is going to haunt you for the rest of your time with microbiology as long as you're with me, right? It allows us to understand some key principles of why bacteria can do the things that they do. So learn it, live it, and love it. All right, so we've already talked about capsules, right? We started talking about sugar and and the ability that some of these bacteria have to form biofilms, all right? And so why do we care about biofilms? And you guys actually answered this for me a little bit yesterday. All right, so that last slide, that's just the exact same slide that you saw yesterday. This one's new though. This is biofilms, and I kind of love this depiction from your book because yes, it's relatively simple, but it highlights a couple key kind of characteristics. So in this case, right, bacteria are out wandering around, right? They're trying to find a good nutrient source. They find a nutrient source here, and they adhere to a surface. Who haven't I asked? Nope, I picked on you. Olivia. Do you remember from yesterday what are two things that we talked about that would allow bacteria to adhere to a surface?
The capsule is one.
Okay.
And then the flagella.
Okay, so capsule's one. I definitely love capsule. Right, flagella is for propulsion. Gloria, can you help her out? What's the second one? Capsule and what?
I'm also looking too.
Okay. Keep looking.
Is it the cell wall?
No. Not quite. I believe.
Can I answer? Is that okay?
If the answer is right, it is.
Fimbriae.
Fimbriae, right? Fimbriae or pili, right? And so bacteria are going to have kind of two major ways they can attach, by fimbriae or by capsules, all right? If they're attaching to a specific surface, right? And so those are kind of two of their main mechanisms. There's other things they can do, but those are kind of two of their main mechanisms, right? So they've attached to their surface. they're really, really happy, right? So they start to divide, right? And they start to produce extracellular polymeric substance, right? EPS. This is going to be that kind of slime-like, sugary, sticky stuff, all right? What's really cool, if we're talking about a pure biologic phenomenon, is that other bacteria can live, all right, in this extra polymeric substance, right? They can live in this slime. So here you have these rods, right? And now here you have these cocci, right? These little circle guys. And what's really interesting is these two, right, can form a nice relationship because they have either different nutrient sources, or what I love about some bacteria is that one bacteria's waste may actually be a nutritional source for a different bacteria, right? So that allows a mixed population to exist within this biofilm, all right? If we're talking about a pathogenic situation, that can definitely make things kind of dicey and kind of tricky, all right? Here you see, all right, these guys continue to grow. The other thing that is beautiful, 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 and we'll talk about this a little bit later, this concept called quorum sensing. Don't worry about understanding that yet. We'll go over that a little bit later. But being stuck in this biofilm and being in close relationship with your best friends allows, right, not only the sharing of nutrients, right, but it allows communication, all right? These bacteria essentially can talk to each other. Now, again, bacteria talking to each other is very different than us talking to each other, but it allows them to share information. This is where we get very concerned because sometimes the information that they share or what they're communicating about is antibiotic resistance or the ability to utilize a different nutrient that they'd never utilized before, all right? So they can essentially communicate, share, or learn with one another. Now, eventually, right, the biofilm gets very large, right? And it kind of, we'll say almost bursts, right? Maybe not 100% accurate, but it opens up and we release other bacteria that will then go on and colonize someplace else, right? And this ensures the survival of the species. Because if you just stay here, even though this is a nice place to live, maybe it does get destroyed. If it gets destroyed, then your species, right, is gone. The ability to leave and find a new area, right, and this process then kind of starts over someplace else. It keeps on going and going, right, allows the survival of these different bacteria, right? So sharing of nutrients, communication, and the one that we kind of didn't specifically mention, but protection. Remember, biofilms make it very hard for the immune system of any organism to target these bacteria, right? So it's protective. So protection, communication, protection. Why am I drawing a blank now? Protection, communication, sharing of nutrients. There we go. I was like, I know I just said it. It's right there in front of me. Those of you that are interested in cancer or tumor biology, the thing that's really fascinating about this is actually, maybe with the exception of different cells coming and living with it, but this is actually the general process that a metastatic tumor finds, right? So a tumor grows to a large size. If it's metastatic, cells leave that tumor, enter circulation, find a nice home to live in, start multiplying, growing, right? Eventually slough off again and go to other places of the body, all right? And so it's very fascinating how closely those two processes or these two processes resemble one another, at least for me, maybe not for you, but I think it's kind of cool. And again, just a side note, I don't think biofilm infections or cancers are cool, right? But from a biological standpoint, I think they're quite fascinating. So here, this is everything that I basically just told you. All right. A lot of bacteria do very well in biofilms, right? We talk about them in infection, but if you've ever walked through a stream on a rock and it feels kind of slippery and slimy, a lot of times that slippery, slimy stuff is actually the biofilm of bacteria that have adhered to the rock, right? So not only is it important for pathogenesis, but biofilms are important for the survival of bacteria just in the environment, right? They're not, biofilms are not inherently bad, all right? This is just a little bit of a chart that I kind of want you to get familiar with, and it talks about some of the different carbon requirements of microbes and the different energy requirements of microbes. So, you know, we could actually put you on this chart, and I'll show you or tell you where you're on this chart in just a second, but you need carbon, right? Because carbon is essential to all the macromolecules of life, right? So the macromolecules of life would be carbohydrates, right? Lipids, right? Amino acids that then become proteins, right? And nucleic acids, right? Those are all carbon based. And so how we get our carbon is essential, right? How we get our energy, all right, is essential too, because our energy, right, the energy that we acquire allows us to build things that we need. And so what this chart is telling you is, it's kind of a two-way chart, right? Carbon going this way, energy going this way. And so you have two options for carbon, right? You either get inorganic carbon, CO2, or you get organic carbon, right? Organic carbon, think things that you're eating, right? A cheeseburger is an example of something that contains organic carbon, or insert your favorite food source here, all right? You can't do this, right? This is for trees and carbon fixing bacteria, all right? Energy source, sunlight. You also cannot do this, right? This is something that's capable of photosynthesis. However, right, this is where you guys can fall in line, right? You either break down certain chemicals or other compounds, and you rely on the energy that is released from the breakdown of those compounds, right? This is you. So we get an example of where you find you are a chemo-organo-heterotroph, all right? You have, right, or you use organic carbon, and you rely on, right, the breakdown of carbon, right, or other chemicals to release energy that you then need to use. This is most disease-causing bacteria. What is the source of organic carbon for disease-causing bacteria? Don't overthink this, please. Caroline, what is it? What's the source of organic carbon for disease-causing bacteria?
Is it in the air?
The air?
In the soil?
For disease-causing bacteria. Where does the bacteria live when it's causing disease?
Oh, in the body.
The body, right? You are the organic carbon source, right, for disease-causing bacteria, right? They need you, all right? The problem is when they start to eat you, then we have some other issues that are going on that are unpleasant for us, right? Here, right, another category, right, photoautotroph, right? So that means, right, photosynthesis and its carbon source, right, autotroph, right, it's pulling in inorganic carbon, right? So photoautotrophs, this is where we're going to find typically our carbon and nitrogen fixing organisms, right? You do have photoheterotroph, which is interesting, right? They're using organic carbon as their carbon source, but they're still able to get energy from the sun, right? So essentially they're getting their energy from the sun, and they're using the carbon from an organic carbon source to help build things. And then you have down here, right, these chemolithotrophs. These are some of these extremophiles that we'll talk about briefly. You can also have carbon-fixing bacteria that fall into this category. So, right, remember, they are using, all right, chemical compounds or chemicals and other compounds as their energy source, right? And then they're using CO2 as their carbon source. And so, you know, this could be a good matching question, potentially. Gloria, did you see that? All right, good. Just want to make sure. No excuses. All right. So a lot of bacteria have a similar tolerance to the environment that we have. All right. You know, we could almost say they're needy, right? We're pretty needy. The conditions that are required for us to live, although you may think you're well adapted and able to survive anything, you know, compared to a lot of other organisms, that's not really true, right? We need a lot of different things. And so some bacteria fall into that same category that we do. Some, however, right, have a totally different set of tolerances. And so this is the group of extremophiles. This is dominated by archaea bacteria, right? That other domain. There are prokaryotic bacteria that can fall into these categories. There are even fungi that fall into some of these categories, right? But we're primarily referring to archaea bacteria with some bacteria kind of looped in there right now. And we'll talk about some of the different cool things that fungi can do and where they can live later. So we talk about extremophiles, right? You guys did this actually a couple days ago when you were looking at some bacterial growth stuff, right? Temperature, right? A whole range of temperatures. pH, right? So really acidic, potentially really basic environments. Salt, right? So we talked about halophiles. You could also even add sugar in here. And then water, right? The ability to potentially be aquatic is really unique. So this is a chart that you guys are probably kind of familiar with. You should recognize some of these terms, right? Psychrophile, psychrotroph, mesophile, thermophile, hyperthermophile, right? So psychro meaning cold, right? So these guys are great at surviving colder temperatures. These, along with fungi, are why even if you leave your food in the fridge, right, it still has a limited shelf life because there are things that are happy to grow in it. This is also an example. If you wanted to, you could go to Antarctica, right, and you can dig in the snow there, and you can actually find bacteria that are living in the snow in Antarctica, right, because they are doing a good job of growing at those really cold temperatures, right? Mesophile. Why do we care about mesophiles?
They grow in like human, like in the range of like human temperatures. So like those are like the kind of bacteria that can survive in like us humans.
Right, perfect, right? So mesophiles, right, are going to contain pathogenic bacteria that we should be concerned about. But mesophiles also contain the bacteria that are part of our microbiome that are really good for us, right? So when it comes to bacteria that's living in us, good or bad, right, they're going to kind of fall in this category. And you can see their optimal growth temperature, right, is almost perfectly aligned with body temperature, right? So they do really well inside of us. You see down here, right, room temperature is about 22 degrees Celsius, right? And so you can see these guys still grow really well there. This is why if you're culturing bacteria, you could leave it out on the bench top, that plate, and it would eventually grow. It would take a lot longer, right, than if you would pop it in the incubator and kind of letting it grow at that exact temperature, right? Thermophile and hyperthermophile, right? These are starting to grow at really, really hot temperatures, all right? If you click on this link, it takes you to an article about this. This is a prismatic spring in Yellowstone Park. And this is kind of like an aerial view. There's a little walkway down here that you can come along to look at it. It's a sulfur hot spring. Temperatures approaching boiling. And the reason that it looks so cool and beautiful, right, is these are actually bacteria that live in here, right, and give the spring its color, right? So these bacteria can live in this really, really hot water. What's also cool, depending on when you go, seasonally, the bacteria kind of changes the dominant population. And so you'll see different colors depending on when you go, depending on which bacteria are the dominant population at the time. All right, so if you've ever heard too of bacteria growing near volcanic vents deep down in the ocean, right? Those bacteria are able to withstand these high temperatures, right? So those are thermophiles and hyperthermophiles, all right? Also, just wanted to point out, if any of you have heard of PCR, polymerase chain reaction, this is basically how we take one strand of DNA and we make a bunch of copies. And so those of you that are maybe interested in crime shows, right, you know, they find a sample of DNA from the victim or from the murderer, right? And in order to analyze that, they need a lot more of that DNA. And so they make a bunch of copies of it in the lab via something called PCR. And during that chain of events, you heat up the DNA and you make it split apart. But then there's a special enzyme in there that's able to function at high temperatures and make new strands of DNA. And that enzyme was actually isolated from an extremophile, from a bacteria that was capable of replicating in really hot temperatures, right? So this is really visually stunning, but these bacteria have also helped us with an essential research tool, right? So, you know, the other kind of extremophile would be something that can grow at a different pH. In here, right, I love these names. They're relatively simple. An acidophile grows in really acidic conditions. A neutrophile grows in neutral conditions, you can see here. And then an alkalophile grows in alkaline, right, aka basic environments. And what you see here on this slide are kind of some common bacteria. Some of them are useful to us. Some of them are pathogenic. Here we go. So Lactobacillus acidophilus, right? This is, those of you that maybe like yogurt, this bacteria is present in yogurt. And so yogurt is actually slightly acidic. Staph aureus, right, which can cause infection in humans. If you notice, it's optimal pH. What do you know? Hey, let's see. Mercy, what's something that you know that has a pH that's about neutral? Mercy? Nope. Okay, maybe not there right now. Amanda, tell me something that has a neutral pH that we should care about.
Blood?
Okay, blood, perfect, right? Blood, I would just go in general and say for the most cases, you, right, have a lot of neutral or sites of neutral pH in you, right? And so this is a bacteria that can cause a lot of problems in us. You also see that down here with Streptococcus pneumonia. Now, you know, how sensitive are bacteria to this system? Well, that's a great question. So this is an example. So the normal, a normal lung, which I assume many of you have, maybe you don't, the pH is around 7.4, right? So, you know, slightly basic. All right. And here you can see Pseudomonas aeruginosa, right, which is a bacteria that can be pathogenic. Its kind of optimal pH is 6.6 to 7.0, right? And so the fact that your lungs are 7.4 is outside of its optimal pH range. Now, it can survive in your lungs, right? But it's not ideal. And so most of us don't have major issues with Pseudomonas aeruginosa in our lungs. However, right, people that have cystic fibrosis, for a multitude of reasons, right, have a lower lung pH, right? In this case, their lung pH is around 6.8. That, right, falls into the optimal range, right, of Pseudomonas aeruginosa. So somewhat cystic fibrosis, right? If you've known someone with cystic fibrosis, if you have cystic fibrosis, right? I don't know that much about you guys, right? People with cystic fibrosis are constantly fighting lung infections, are much more prone to lung infections, are worried about lung infections. And one of the reasons is their difference in their lung pH allows, right, for the optimal growth of pathogenic bacteria. All right, so this is one of the reasons that they can have a multitude of issues. All right. This is one of my favorite. All right. Salt and sugar. If you remember osmosis, right, water is drawn to high concentrations of solute. Right? That could be salt or sugar. That means that salt or sugar, right, if you're talking about the two pictures you see here, are great preservatives. And they're great preservatives because beef jerky is very salty, jam is very sugary. And what happens is that salt and sugar attracts water. And when that salt and sugar attracts water, it pulls it away or keeps it from bacteria. All right. Now, there are, right, and you guys identified these, some organisms that do very well, right, in salt. So you have halotolerant, right, organisms that can survive in high concentrations of salt. It's maybe not their priority, right? So I think I was talking to some of you, and I think it was referring to, you could also refer to this as a facultative halophile, right? So remember, facultative means that it's not required, even though there may be a preference for that bacteria. You have halophiles, right, which require salt, right? So you guys could also call these, right, obligate, right? Obligate meaning that it needs it, it requires it. Usually around 3% salt concentration. And to just give you an idea of how salty that is, ocean, right, water or seawater, if any of you like the beach, is around 3% salinity, right? And so that's pretty salty. So you can imagine something that can live in 10% salt. And then you have your extreme halophiles, right, that live in salt concentrations that are 9% or more, right? Incredibly salty. One of the plates that you guys will encounter in some of your lab activities is something called a mannitol salt agar plate, an MSA plate. And it has a salt concentration of around 6.5 to 7%. And it is actually great at growing bacteria that we find on our skin, specifically Staphylococcus species. And so your skin, right, is a relatively salty environment, and the salt on your skin actually helps regulate or control what bacteria can grow on your skin. All right, so kind of cool there. I would be, I think, irresponsible. We didn't talk some about oxygen requirements, and this, will kind of pop in every now and again throughout the semester about oxygen. And again, this is a great opportunity to introduce those words, even though I mentioned them, obligate and facultative. So an obligate aerobe is something that requires oxygen, right? If oxygen is not present, these bacteria will not survive, right? You then also have a microaerophile, and these are kind of fascinating because they have a very specific range of oxygen. They like 2 to 10%. Anything below 2%, anything above 10% is considered toxic, right? A great example of a microaerophile is H. pylori. Does anyone know what H. pylori is associated with? Why we care about that?
Is that ulcers?
Yes, right. This is the bacteria that causes stomach ulcers. All right. And so it wasn't until the 1980s, right, that we realized that one of the best ways to treat ulcers in a lot of patients is actually to give them antibiotics. All right. 2 Australian scientists figured this out and actually got the Nobel Prize for it. So it was a big deal, right? Facultative, aerobe, right? Best with O2, but they don't need it. You could also call this a facultative, anaerobe. I'm fine with those being used interchangeably, right? Because it's just saying, hey, right, they may like this, but they don't need it. All right, so that's kind of what facultative is saying. You then have obligate anaerobe. So any oxygen is toxic. So Clostridium botulinum. So if you've heard of botulism, does anyone know why botulism used to be such a big problem, or where it caused a problem?
Isn't it like in cans if they were dented?
Well, okay, so yes, definitely with cans. I'd have to look up and see how true it is about dented cans, but we didn't quite have a great understanding of the canning process. So now when things are canned, they're actually like pasteurized, just like you would milk. And so what would happen is you can something, you remove the oxygen from that can, and then you'd make the perfect environment for Clostridium botulinum. And then someone would ingest, right, that canned product and they die, right? Botulinum messes with your muscles, all right? And one of the muscles you don't want things messing with is your diaphragm, right, which is important for breathing. And so this could cause death in patients. If any of you have heard of Botox, right? Botox is used by some folks as a way to, I guess, beautify themselves, right? Is what they would say. Botox is also now being used for migraine treatments, all right? And so we can actually utilize the toxin for good things. Well, I don't know if Botox is a good thing, but we can utilize it to our advantage, but it can be very dangerous when ingested. But now the modern canning process is not really an issue anymore. I'd have to check on dented cans. My wife always tells me not to pick the dented ones also, but I don't know if it's because of botulism. Rachel, you can double check that for me, okay? A little side research project. Okay, I expect an answer. All right, aerotolerant anaerobes. These are bacteria that don't utilize O2, right, but are fine in the presence of it. All right, they just kind of go with it. It doesn't hurt them, but they're not utilizing it. An example of this would be S. pyogenes, which we would have used in the lab if we had been there, but we aren't. So just kind of know those different kind of categories of oxygen requirements. All right. One of the things that we'll talk about a little bit more, especially as it pertains to your projects, because one of the tests you guys may want to do for your lab project, and again, we'll talk about that probably on Monday, is something called a catalase test. And what we see here is that if oxygen is present, it results in the generation of reactive oxygen species. So some reactive oxygen species that you may have heard of would be hydrogen peroxide and superoxide, right? Those are kind of the two of the big ones. And hydrogen peroxide and superoxide can damage cells in a number of ways. They can interfere with proteins, they can interfere with the membrane. They're not a good thing to have present in large amounts, all right? And so if you are capable of aerobic respiration, right, you have an electron transport chain, that means that more than likely oxygen is your terminal electron acceptor. If that's the case, then you're generating these reactive oxygen species. So you have to have a way to combat them. So if you've seen some of these supplements that claim to be antioxidants, right, that's what they're going after. They are saying this has a compound or a chemical that helps combat these reactive oxygen species. All right. And so kind of two of the big ones are SOD, superoxide dismutase, right? Superoxide dismutase takes superoxide, turns it into hydrogen peroxide. And then you have catalase, which takes that hydrogen peroxide and turns it into water. And then you'll also see some oxygen produced as a result. And so when you do this test, you'll actually see a bubbling, and that's because of the oxygen that's being produced, right? So this is neutralizing those reactive oxygen species. Now, I hate to burst your bubble, and I'm not saying don't eat healthy, and you know, a lot of foods that have antioxidants in them are really good for you. And I wasn't in grad school that long ago, but up to that point, there had been no treatment with antioxidants that was able to reverse a pathology, right? Was able to cure a disease. The amount of these antioxidants that you would have to take in to combat a pathogenic situation or even some genetic abnormality is almost impossible, right? But people continue to look at them, and maybe they'll figure out how to utilize them later. So some microbial growth nutrients. We've already talked about carbon and oxygen, right? You can throw hydrogen in there because hydrogen and carbon, right, when they're together, that's what forms organic compounds. So amino acids, lipids, nucleic acids, and sugars, right? Those macromolecules. Nitrogen, right? They need it for amino acids and nucleic acids. Sulfur, right? Found in some amino acids. Phosphorus, you can see ATP, membrane lipids, nucleic acids, potassium, magnesium, and calcium, right? Required for certain enzymes. And a lot of these, at least in us, are important for some signaling mechanisms. And then iron, right? Part of certain enzymes, just like we mentioned for cellular function. So hopefully when you look at this chart, you're like, huh, Professor Daft, those are the same things that we need. Boop. Bingo, right? And so this is why I chose that iron paper, right? It is highlighting this idea of the same things that they find essential are often the same things that we find essential. And so it becomes a competition for the same nutrients, all right? And sometimes that competition can be a little fierce, right? We already talked about carbon, right, as a source, but again, this is kind of breaking down that table a little bit and just looking at the carbon source. So a heterotroph, right, is using organic carbon, right? You are a heterotroph. Pathogenic bacteria are heterotrophs. An autotroph, right, is using inorganic carbon, aka CO2. These organisms, right, are capable of carbon fixation. They are taking that atmospheric carbon and they are actually converting it to organic carbon, right? Bacteria can do this, or some bacteria can do this. And this is what you have going on in plants, right? Plants are taking in atmospheric carbon, aka CO2, and turning it into organic carbon. Nitrogen, right? We just mentioned nitrogen and phosphorus on that last chart. All right. You know, when we talk about bacteria and nitrogen, this is really important because bacteria can take atmospheric nitrogen and make it into ammonia, which then can be taken up in plants, right, and incorporated into their macromolecules. And then we eat plants, right, or we eat things that eat plants. We talked about phosphorus and iron, right? Or we talked about iron specifically, we can throw phosphorus into here, right? I say necessary and limited. And so the quantities or the amount that are available, right, of things like phosphorus and iron dictate the size of, if we're talking about bacteria, the size of that population, right? If there's an abundance of iron or phosphorus, then there's going to be an abundance of bacteria because they're going to grab onto it and grow like crazy. If the amount is limited, right, like you saw what your body is trying to do, right, through things like lactoferrin, ferritin, right, siderocalin, right, hemoglobin, myoglobin, right, your body has ways to hold on to iron. If you keep iron and you limit the amount that's available, well, then you limit the amount of bacteria that can grow as a result of its presence. So, this is one of the negative things about cleaners, right? Some cleaners are phosphate based. Even fertilizers contain a lot of phosphate in them. And when you use these in large quantities and they run off or they pool or collect in the same areas, you can actually have bacterial blooms, right? Rapid growth of a certain bacterial population. So sometimes if you've seen situations where there's been a fertilizer runoff, maybe from like a big hog farm into a local water source, and then within a few days you have a bunch of dead fish, right? And the reason you have a bunch of dead fish is because the bacteria in that water source took advantage of all of that extra nutrients they grew, right? Caused infections in the fish, right? And you can kind of have a cataclysmic event there. All right. So those are nutrients, right? When we talk about growth factors, all right, this is something that is needed but cannot be synthesized, right? So that means that it needs to bring it in from the outside environment. So an example of this would be an essential amino acid, right? And you guys, you have 20 amino acids, right? And you can synthesize almost all of those amino acids from other components of macromolecules, right? So if you don't have it, you can build it. There are some, right, essential amino acids that you have to ingest. You have to take in from the outside environment, right? And so bacteria, just like us, have that requirement. There's not everything that they can synthesize, so sometimes they need you to provide it for them, all right? We've talked about energy, phototrophs, sunlight, chemotrophs, chemical compounds, which includes organic carbons, right? Again, that's going back to that chart. Okay. So, stop that share there. That's good. Oh, I want to. All right. So this chapter is on metabolism, right? A lot of components of this chapter are almost identical to the metabolism that we see in microbes, right? And this is why a lot of microbes are such great research tools, because they possess a lot of the same metabolic pathways. We will point out some differences, but you'll see there are a lot of similarities, right? So how are we utilizing carbon, right? And carbon's a great energy source, or organic carbon, right? Molecules are great energy sources. We'll talk briefly about enzymatic principles. We'll talk about the central metabolic pathways. That's going to be a big part of this chapter. And just kind of briefly go over, even though I think some of you have had this before, anabolism, catabolism, respiration, and fermentation, right? And again, we're not going to get to all of this in the next little bit, right? But at least we'll start. So when we talk about metabolism, right, we're talking about the sum of all chemical reactions in a cell, right? So metabolism is essential for harvesting energy, all right, converting it into a form, right, that can then be utilized, right, and that energy will be consumed, right? So this is the idea of catabolic reactions and anabolic reactions. In this case, right, we're talking about us, right? We break things down to release energy. We then use that energy to build things that we need, all right? And as I mentioned before, a lot of metabolic pathways in microbes are similar to those in humans. This is a chart that I actually shown in my A&P1 class. This is every metabolic reaction that takes place in the human body. All right, so you could zoom in. I don't know if the quality of this image is enough, but you could go find this. This is by a group from University of California, San Diego. And it just shows you, right, there's a lot of chemical reactions going on in your body. And bacteria are going to have a very similar chart, right? So basic principles, right? Catabolism versus anabolism. And I kind of like this because it shows you that they are feeding into one another. Now, for catabolism, right, you are releasing energy, which is essentially really, really essential. You are also, right, releasing precursor metabolites. And what that means is when you break down a larger molecule or compound, all right, portions or pieces of that molecule or compound could then be used to build something else, right? And the energy that you're using is coming down maybe from that, even that same breakdown, right? So you break down to get energy, and you can see here some of the things that that energy is going into, right? Break down, energy, and then you build back up. Okay, and we'll talk about some of these precursor metabolites, and we'll talk about this energy that's being generated as we kind of go forward. All right. So again, you guys did some of this in your chemistry, I think, review. I think I had some questions related to this, but maybe you've heard of redox reactions, right? That is a oxidation reduction is what that stands for. Remember, Leo goes Ger, loss of electrons is oxidation, gaining electrons is reduction. And basically what that is saying is that biology and chemistry are not magic, right? When you break something down, it goes someplace. Things don't just disappear. There's something there to pick them up. And so redox reactions, right? If you've heard the electron transport chain, which we've already mentioned once or twice, and we'll go over more, we probably won't get to it today. The electron transport chain, right? Passing those electrons along, that's a series of redox reactions. Something accepts the electron, something gives away the electron. Something accepts it, something gives it away, right? This is is essential to life, and for us, essential to the generation of energy. I don't expect you to memorize all of these, but I do want to point out a couple important things. Here are some precursor metabolites. All right. These are generated when you are breaking down organic sugars. All right, and you can see the pathway that they're generated in. So you probably, guys, hopefully to this point, have maybe heard of glycolysis. If not, again, we're going to go over it. The pentose phosphate cycle is maybe one that you're not quite as familiar with. We'll go over that. The transition step occurs between glycolysis and the TCA cycle and the TCA cycle. All right, and so as you go through these pathways, right, you're breaking things down, and these are some of the things that are generated during the breakdown. And over here, you can see what these precursor metabolites can be used for. All right, so if you look down here, right, oxalacetate can be used to make proteins, right, because it's used to make these different amino acids. Here you can see dihydroxyacetone phosphate, which is generated during glycolysis. What it helps make lipids because it's part of glycerol, right, which is an important component of a lot of lipids, right, or is an important component of lipids to have a glycerol head. Here's something, though, that's kind of unique, right, and glucose 6-phosphate and fructose 6-phosphate are not unique metabolites, right? You generate these when you're going through glycolysis, but, right, glucose 6-phosphate and fructose 6-phosphate can be used, right, to make lipopolysaccharide and peptidoglycan, right? These are not present in your cells, right? So here, right, is kind of a unique difference. Some of the precursor metabolites that are generated from the central metabolic pathways that we're going to go over in more detail, right, are being used to produce or synthesize some of the important components that are unique to bacteria that we do not see in our cells, right? You do not have LPS, that lipopolysaccharide. You do not have a cell wall. You don't have peptidoglycan, right? So you are not able to utilize those precursor metabolites for the exact same things. You look down here on this chart, a lot of these things, right, are almost identical between us and bacteria, but you see up here, right, there are some kind of unique differences. And again, these precursor metabolites are used to build, right? They are used, I have it right down here, in anabolic reactions. All right, the central metabolic pathways, let's kind of jump right in. They're going to be glycolysis, pentose phosphate pathway, right, and the TCA cycle, all right? Again, you can call the TCA cycle by a couple different names. I don't care which one you call it by, as long as you're referring to the right cycle, all right? So the point of the central metabolic pathways is to generate some ATP, right? We call this substrate level phosphorylation. Basically what that means is that you have an incomplete breakdown, of your organic carbon source. You haven't finished the job, and I'll kind of give you an example of that here in just a second. The most important thing, everybody look right at me right now. The most important thing of the central metabolic pathways, and I said this in my ANP1 class this last semester, so some of you should know, right, is the generation of reducing power, all right? And reducing power exists in the form of NADH, FADH2, right, and even NADPH. NADH and FADH2 are going to travel to, eventually, if you have it, the electron transport chain, and result in the generation of a lot of ATP. The central metabolic pathways themselves do not directly result in the production of very much ATP. You need the reducing power that they generate in order to get a bunch of ATP. And then, as we already mentioned, right, these precursor metabolites are being generated by these pathways that can then be used in anabolic reactions to build up different things, right? So far, right, we are kind of running almost identical to the metabolism that is occurring in prokaryotic cells and eukaryotic cells, right? With the exception being so far glucose 6-phosphate and fructose 6-phosphate, right? Being used for either the production of LPS or for the production of peptidoglycan, right? But right now we're running kind of similar, right? Along a similar parallel path, right? Sharing some of these same pathways. This is just kind of a quick summation, and somewhere in your textbook you have a similar figure, but it's just showing, right, how these pathways are linked. So this is kind of a good starting point. One of the things that I want you to note is that this is not a linear progression. You don't start glycolysis, then go into the pentose phosphate pathway, and then go into the TCA cycle, right? What it's showing here, and I love this image, is it's showing glycolysis does feed into the TCA cycle. But the pentose phosphate pathway you kind of have here out to the side, right? It's not necessarily following that sequential order, right? You also see here, and we'll talk about these in more detail, you can actually divert from glycolysis to fermentation, all right? And, right, you can go from these different pathways eventually, if oxygen is present, we'll talk about some exceptions to that, and intercellular respiration, right? So this is a really good chart to start with. It doesn't have a lot of detail, but it does show how these metabolic pathways can be linked. Please remember, when we refer to central metabolic pathways, we're talking about glycolysis, pentose phosphate, and the TCA cycle, right? Cellular respiration is essential, is very, very important, as long as there is oxygen present or another terminal electron acceptor, right? But we kind of consider it a separate event from these metabolic pathways, even though they are feeding into it, okay? So, what are some of the key molecules, right? So glucose is probably one of the most important, right? And I say other hexoses, so other 6-carbon sugars. Glucose is a sugar source that's easily utilized by almost every cell out there. If glucose isn't present, a lot of cells are going to convert other molecules into glucose, all right? These metabolic pathways are regulated by enzymes. Any step that occurs is going to have an enzyme that is associated with it. Pyruvate and acetyl-CoA are essential to the central metabolic pathways, right? Pyruvate is the end product of glycolysis. Acetyl-CoA is the starting product of the TCA cycle. And as I mentioned before, NADH, NADPH, and FADH2, right, are going to be generating that reducing power, which eventually will lead to the production of ATP. Or if we're talking about NADPH, we'll talk about what it does. It's a little different here in just a bit. And then finally, you can see I have at the bottom of the list, and I don't even think I did this on purpose, but I hope it emphasizes that the central metabolic pathways, their direct production of ATP is rather limited, right? It is NADH and FADH2 that are generating the production of most ATP. We'll talk about NADP. It's a little different, but we'll get back to it. Okay. All right. So again, we're not going to spend a lot of time on this. I don't know how comfortable you are with enzymes. I'm not going to go into a ton of detail, but I do want you to remember that enzymes are organic catalysts, right? That means they start chemical reactions. They move relatively quickly, right? They're rapidly acting. They regulate all cellular activities, and they're highly selective, right? And so if we're going back to chemistry 101, you have compound A, you have compound B. If you are waiting for compound A and compound B to find each other naturally and come together, it's going to take a long, long time, and perhaps even a lot of energy to make that happen. Enzymes are important because they can bring those two, they can bring A, they can bring B together, and they decrease the amount of energy that's required for those compounds to join with one another. Or you could even say the same thing about breaking something apart, right? So enzymes, right, are part of every process of the central metabolic pathway. Now, glucose, right? And sometimes you'll see it in its hexagonal form, right? Becomes pyruvate. That is kind of what you start with, and this is what you end with, right? Simplified. You start with a 6-carbon molecule, you end with 2 3-carbon molecules, right? I'm only showing 1 pyruvate here, but there are 2 pyruvates that are generated. All right. This is a little bit more of an in-depth look at glycolysis. All right. Here you can see, right, some of these different precursor metabolites that were shown a few slides ago. All right. But here the same idea, 6-carbon, and then you end with 2 3-carbon pyruvates. Now note, if you decide to use glucose 6-phosphate, fructose 6-phosphate, if you're a bacteria, even if you're one of us, this dehydroxyacetone phosphate, if you use any of these as a precursor metabolite, then you are taking away, right, your ability to generate energy, right? Because if you're using them to build, well, then you can't continue to break them down to eventually get energy out of it. Can you tell me, right, or can someone tell me what the condition of a cell would be, whether bacterial or whether your cell, that would allow you to divert one of these precursor metabolites to an anabolic pathway? What would allow a cell to do that? What situation would it have to be in to do that? Don't overthink it. Please keep it simple. Anyone?
If it was like overwhelmed with like glucose and energy.
Okay, I don't know if I'd use the term overwhelmed. I would use if there is enough ATP present, right? If ATP is abundant, if it has enough energy, it doesn't need any more energy, then it can start diverting things to anabolic pathways, right? If you're low on energy, well, then you're not going to divert precursor metabolites to build things, right? It would be like if, Lauren had $0 in her checking account. She would not go out and start building a house, right? Houses cost money. Now, if Lauren had $500,000 in her checking account, right? She's got money in the bank. She can start building her house, right? She can make that investment. And so one of the things that I try to hammer home to my students, no matter what class they're in, if we're talking about metabolism, is the energy status or energy state of the cell is going to determine which pathways are being utilized and how they're being utilized. If you have a lot of energy present, fine, you can start to divert to anabolic pathways. If you are lacking energy, well, then you're going to have to hold off on the anabolic pathways, focus on the catabolic until you have enough energy to build or do the things that you want to do. All right. We're going to finish up with glycolysis, and then that'll be as far as we get here. All right. So for glycolysis, right? There's a couple different phases here. And if you look in your textbook, it'll talk about these. You have an investment phase and you have a payoff phase. In this investment phase, you're actually spending money, right? You're spending the currency of cell energy. You're spending ATP, right? And you do this when you're phosphorylating glucose, which eventually is becoming fructose, right? But you then have a payoff phase. And the payoff phase is the generation of 4 molecules of ATP and 2 molecules of NADH. Now, this is, again, where I emphasize this point, glycolysis, right? You are only generating 2 ATP. This is not a high yield of energy. Where your investment, right? And I know you have 2 phases, so let me change my word there. Where your real payoff, there we go, is coming from is the generation of NADH, which eventually will travel down to the electron transport chain and result in the production of more ATP, right? So please, 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 for any of the central metabolic pathways, I do not mind if you mention ATP. Well, I would mind if you mention ATP and pentose phosphate. We'll get there later. But if we're talking about glycolysis or the TCA, if you mention ATP, that's totally fine. But please, please, if you don't mention these molecules, right, that are reducing molecules, you're going to be in trouble on my exam, right? I would say my red pen will come out, but it won't, because I won't be grading you guys with a pen, right? But my, I guess, delete button, I don't know, typing fingers, whatever will come out, right? Reducing power, reducing power, reducing power. So that's it, right? We're going to stop here. What I will do is we'll pick back up on Monday with the rest of this. You guys will do your exam. Speaking of exam, what I'm going to do, and this helps some of you maybe more than others, but I am going to have the exam start at 9, all right, Eastern time. So for those of you in Washington, it'll be 6 instead of 5, right?
A little bit.
Huh?
Kind of helps.
Yeah, it kind of helps a little bit, right? See? So we're going to start the exam at 9, all right? And I know there's a few of you that have reached out to me that have some extended test time. I guess requirements or benefits there. And so for the regular exam, right, it's going to be from 9 to 10:30, right? So you have an hour and a half to complete the exam. For those of you that have extended time, right, I will give you a little bit of extra time, right? And extended time, I know a lot of you want that. This is extended time via academic services, right? But I typically, give my students somewhere between an hour and 75-ish minutes to do these exams. And so I'm actually even giving everybody else a little bit, more than that. So please be ready to go at 9, right? That's when I'll open it up for you guys. All right? Cool.
Dr. Daft?
Yeah?
Yeah, so I'm going to be working on the ambulance tomorrow, and shift starts at 9. So what can we do about that?
You can take it earlier.
Yay.
Yeah.
Is there a possibility that I can do it either on like Saturday and it open up then, or late at night? I don't know. Like a get off shift.
I'll think about it. I'll come up with some type of solution. But with that being said, it is about 10:10. So let's take a break until 10:25, and we'll come back together and we'll see if maybe I can get you into your breakout rooms again. If not, then we'll just adapt and survive, right? So let's take about a 15-minute break. So 10:25, I'll expect